Assalamu alaikum students. Today we are going to start our lecture, and in this lecture we will discuss our last two long questions, which was long question number 7 and long question number 8. So our first question is question number 7, which states that how biological method is applied to find the cause of malaria. So before we get into details, let me ask you something. Have you ever seen someone having high fever with chills and sweating? Or maybe someone in your family got sick after mosquito bites? That disease could be malaria, which is very common in many areas, especially where there is dirty or standing water. Malaria is a serious disease that has affected millions of people. Scientists studied malaria as a biological problem and used the biological method to find its cause. So, in this question, we will learn how scientists discovered the cause of malaria using the biological method. The biological method is a step by step way to solve problems in science. Okay, so let's see how it was used for malaria. So, starting off with the symptoms of malaria, let's first understand the symptoms. A malaria patient usually shows feeling of very cold and shivering, okay, like chills feeling. Second is high temperature fever above normal temperature and headache, feeling nausea, like having vomit or sweating, after which the patient feels a little better. And this cycles repeat after 24, 48, or 72 hours. So if you see this pattern, it is a strong sign of malaria. Next is the second big question, which is what causes malaria? By using the biological method, scientists discovered that malaria is caused by a tiny organism called plasmodium. As you can also see in the figures, these are plasmodium. This organism lives in the blood of infected peoples or infected patients. So now let's see how scientists actually found this out by step, okay? So first is the recognition of the problem. So first scientists identified the problem. So first step is the recognition of the problem. So malaria had been affecting people since ancient times, but its cause was unknown. Okay, students. So our next step is observations. So, in the 19th century, scientists made several important observations. First one is that malaria was common in marshy areas. Second is that a drug called quinine helped treat malaria. Third one is drinking marsh water did not cause malaria. Fourth one is a microorganism called plasmodium was found in the blood of malaria patients. These observations helped scientists move closer to the real cause. Next step of biological problem solving is hypothesis. In 1882, a French doctor named Alphonse Laveran studied malaria patients. After his observations, he made a hypothesis. A hypothesis is a smart guess. His hypothesis was Plasmodium is the cause of malaria. Plasmodium is a tiny parasite which was the cause of malaria. Next step is deduction. From this hypothesis or guess, scientists made a deduction. A deduction means if this is true, then this should happen. Okay, students? The deduction was if plasmodium causes malaria, then every person sick with malaria should have plasmodium in their blood. So it's a condition, which means if this is true, then this should happen. Okay, so deduction means this. Next step is experiments. To test this idea, scientists did an experiment. They examined the blood of 100 malaria patients under a microscope. To make the test fair, they also checked the blood of 100 healthy people as a control group. Next is results. The results were very clear. All 100 malaria patients had plasmodium in their blood. Out of 100 healthy people, 93 had no plasmodium in their blood. Only 7 healthy people had plasmodium in their blood. But why these 7 healthy people have it? Scientists found that plasmodium was in the incubation period in their bodies. So what is incubation period? It means the time between when a germ enters the body and when symptoms appear. After a few days, these 7 people also got sick with malaria. So the result proves that hypothesis correct. Plasmodium is the cause of malaria. Sixth step is the reporting of the result. Scientists share these results with the whole world. This helped doctors and governments understand malaria better and find ways to control it. So, students, remember the biological method has clear steps. Starting with problem, hypothesis, deduction, experiment results, and reporting. Using this method, Laveran proved that a tiny parasite called Plasmodium causes malaria. Okay, students, so our next question is that explain use of biological method to understand the spread of malaria. So, How does Plasmodium get into our blood? Today we will see how scientists use the biological method to prove that mosquitoes spread malaria. Step 1 is the recognition of the problem. Malaria has been a deadly disease for centuries. Scientists already knew that Plasmodium causes malaria, but they did not know how it enters the human body. They observed two important things. Malaria was common in marshy, swampy areas, but drinking marsh water did not cause malaria. So the conclusion was simple that Plasmodium is not in the marshy water, it must be carried by something that lives near marshy water. The problem was, what is that carrier? So, second step is observation. In 1883, an American scientist named AFA King made 20 observations. Let's look at three top ones. First one is people who slept outside got malaria more often than people who slept inside. Second is people who slept under fine nets were safer from malaria than those without nets. Third one is people who slept near smoky fires usually did not get malaria. All these observations point to one thing, something in the air that bites us and can be blocked by nets or smoke. That something could be mosquitoes. Next step is hypothesis. Based on these observations, King made a hypothesis. A hypothesis is an educated guess. His hypothesis was mosquitoes transmit plasmodium, so they are involved in the spread of malaria. Okay, so next point is deduction. If this hypothesis is true, then some things must also be true. They are called deductions. So, deduction 1 is plasmodium should be present inside mosquitoes. Deduction 2 will be a mosquito can get plasmodium by biting a person who has malaria. Step 5 is experiments. To test this, a British army doctor named Ronald Ross did important experiments in India in 1897. First, he let a female Anopheles mosquito bite a patient who had malaria. After a few days, he killed the mosquito and checked its stomach. He found plasmodium growing inside the mosquito's stomach. This proved deduction 1. Next experiment is Ross did an experiment on birds, specifically sparrows. He let a female Culex mosquito bite a sparrow that had malaria. Then he let the same mosquito to bite healthy sparrows. After a few days, the healthy sparrows also got sick with malaria. This showed that mosquitoes can pass the disease. Later on, an Italian biologist did a similar experiment with humans. He let an Anopheles mosquito bite a malaria patient. After a few days, he let the same mosquito bite a healthy man. The healthy man later got malaria. This proved that mosquitoes really do spread malaria from person to person. So you can also see in the next figure that how it will transmit from the patient to the healthy person. Using the biological method, scientists proved that mosquitoes are the carrier of malaria. They carry plasmodium from a sick person to a healthy person, then they bite. So the biological method follows these steps: problem, observations, hypothesis, deductions, experiments, and conclusion. So this is all for today's lecture. Thank you for listening. Or if you have any query, you can contact me directly. Okay, students? Allah Hafiz.